So far, we've gone over an introduction to biotechnology and how it emerged from the pharmaceutical industry. Now I'd like to take a step back and cover the knowledge and skills which led to the development of biotechnology. You don't need to know what all these terms mean. I'll be defining them as we go through. But as you can see, the seeds were planted in the 1980s and go forth several decades. The process I use here is looking at the Nobel Prizes which were granted for advances which led to the development of biotechnology. So you'll see a bit of a difference in the timelines between the last slide and this one because Nobel Prizes tend to be awarded several years after the research actually occurred. But we see that the first Nobel Prize we look at here is in 1958, which went to Frederick Sanger for determining the protein sequence of insulin. Frederick Sanger actually got another Nobel Prize for figuring out how to sequence DNA, and we'll cover that as well. But it was knowing the sequence of human insulin which enabled Genentech, which we mentioned before, to develop recombinant human insulin, which was the first biotechnology drug. The 1985 Nobel Prize in Chemistry went to Herbert Hoffman and Jerome Karl because they laid the foundations for the development of X-ray crystallography, which allows you to determine the 3D structure of a molecule. This aided in the discovery of the structure of DNA and also enables rational drug design. The 1962 Nobel Prize in Physiology or Medicine went to James Watson, Francis Crick, and Maurice Wilkins. It's probably one of the best known Nobel Prizes in the biological sciences and is for their work in discovering the structure of DNA. Knowing the structure of DNA enabled elucidation of the mechanisms for the control of gene expression and the hereditary transfer of genetic information, and is really at the base of molecular biology. We'll be covering that in a future video. The 1968 Nobel Prize in Physiology or Medicine went to Robert Holley, Hargobind Khorana, and Marshall Nirenberg for deciphering the genetic code and also figuring out how it is translated by cells. Once again, we'll be covering this in the video on molecular biology. The 1978 Prize in Physiology or Medicine went to Werner Arber, Dan Nathans, and Hamilton Smith for their discovery of restriction enzymes and their application to the problems of molecular genetics. This enabled Stanley Cohen and Herbert Boyer to develop the methods to splice genes, which is at the base, once again, for most biotechnology applications, and it led to the founding of Genentech and their development of the first biotechnology drug. The 1980 Prize in Chemistry went to Paul Berg for his study of the biochemistry of nucleic acids, specifically with regard to recombinant DNA, and Walter Gilbert and Frederick Sanger, once again, for their contributions to do with sequencing DNA. The ability to sequence DNA was central to the Human Genome Project. We'll be covering that as well, but basically there would be no molecular biology if there wasn't the ability to sequence DNA. Finally, the 1993 Nobel Prize in Chemistry went to Carey Mullis and Michael Smith for their development of the polymerase chain reaction and site-directed mutagenesis. PCR permits production of many copies of a specific DNA sequence, even if it's in a complex mixture. And so when you look at forensic applications, for example, taking a DNA fingerprint from a crime scene, it usually uses PCR to amplify that specific sequence and then tie it to an individual. It has applications also in paternity, in heritage testing, medical diagnostics, in archaeology, and in anthropology as well. Site-directed mutagenesis is a derivation of PCR, and it allows you to make specific changes to DNA and make alterations to genes as you need them. And so in summary, these factors led to an understanding of the role of DNA in programming the abilities of individual cells. They also led to knowledge of how information is encoded in DNA, the mechanisms by which cells use that information, and also the development of techniques to manipulate DNA and thereby reprogram cells